So in the next we're going to look at how tetrads are made when genes are linked. Okay. So we're going to talk about Lu, but now we're not going to talk about Met because we've already said in the previous example that they're unlinked. We'll just think of another one, say for example arginine. All right. So you have two strains. One might be Lu minus arginine plus, and another strain which is Lu plus arginine minus. Right. So unable to produce arginine, doesn't grow on arginine minus media. You cross those together, but now let's say that these are linked to one another. Okay, so instead of a blue and a red chromosome, there's just a blue chromosome like this. Okay, so this is the sort of beginning of meiosis after S phase. And so if they're linked, then you're going to have Lu minus from this particular haploid strain, and then R plus over here. Okay, because that's the replicated two sister chromatids about ready to undergo meiosis, the parental strain haploid genome, right? And then the other parental strain haploid genome, which would be Lu plus arg minus arg minus. Okay. Now, if these genes are linked, notice something, all right? Remember what we said is the way that you produce non-parental ditypes is that these basically segregate independent of one another. Okay. Now clearly that's not going to be the case, right? So if they're closely linked, every single time you will bring Lu minus with if they're very closely linked, or most of the time they'll carry arg plus with it. All right. So the types of spores that will be common in this particular arrangement, if they're closely linked, all right, would really be one kind, right? The really common ones, right? They're going to be Lu minus arg plus for one set, and then Lu minus arg plus for the other set, and then on the other side, Lu plus arg minus Lu plus arg minus. Okay, now you should be able to tell me if this is a parental ditype or non-parental ditype. Well, there's two kinds of types, and they're of the parental type. All right, so in this particular case, we'll call it a parental ditype. Now, how would you create, this is a parental ditype, how could you create a non-parental ditype in this particular case? Well, you have to have crossing over clearly. You have to have actually two crossovers, right? Because if you put a crossover right here, right? An easy way to think now about this is just think of the haploid spores that arise out of each of these particular final segregations of meiosis 1 and meiosis 2. I'm not going to draw tetrads anymore, but one of those spores would be Lu minus arg plus. Another spore would be Lu minus arg minus. Another spore would be Lu plus arg plus. And then the other spore would be Lu plus arg minus. Now, what type of spore is that? That's a tetratype, right? You have sort of two of the parental types and two of the recombinants inside that tetrad. So that's not going to produce a non-parental ditype. That's going to produce a tetratype. How do you get a non-parental ditype? Well, the only way to do that is to allow both of them to sort of become unlinked, to become crossed over. All right. So you have a crossover between these two chromatids, and you have a crossover between these two chromatids, and that will then produce a genotype. And again, if you just sort of follow the crossovers, Lu minus arg minus, Lu minus arg minus, Lu minus arg minus, Lu minus arg minus, all right, and then the pluses now together, Lu plus arg plus, Lu plus arg plus. And that's not going to be very common. So you have to have a crossover. You actually have to have two of them, and they have to be among four of these chromatids. And this is what is called a four-strand double crossover. And in this particular case, it produces tetratypes. Non-parental ditypes. And other crossovers will produce tetratypes and such. And we'll talk about that shortly. But the critical thing here is that this is rare. So the final takeaway here is that when they're linked, it's hard to produce non-parental ditypes. It's going to be a rare type of case. So in that particular case, the numbers of parental dye types is going to be greater than the non-parental dye types, and typically that's a lot greater.